guess the day? Still on 17.1.
No, but do you remember the one in 3? The menu dude? Yeah, I was in a restaurant.
Okay. So we have these restriction enzymes. This one called EcoR1 cuts DNA at GAATTC. Here we have our 4 suspects for the crime. We got their DNA. We just had a small segment of their DNA. We didn't get the whole thing. We just cut out a variable region and copied it many times. How did we do that? Do you remember how we did that? The process for getting these 4 variable regions of DNA copied?
Tactful
what was the whole process called?
PCR.
PCR, polymerase chain reaction. We got pieces that we're interested in, and we cut these pieces with the restriction enzyme GAATTC. They're also called restriction endonucleases. Can you say endonuclease?
Endonuclease.
All at the same time?
Endonuclease.
Chandler, you even tried to say it. So this thing will go along the DNA until it sees GAATTC, and there is no GAATTC sequence here, so that will not be cut. There is a GAATTC sequence here. Snip, snip, cuts it. There is also a GAATTC sequence right there. Snip, snip, cuts it there. There's another one here. Snip. And so this one gets cut in 3 pieces. And this one has 1, 2, 3 GAATTC sequences. So it'll fall apart into 4 pieces. We then take a gel, just like we did last week. We cut little wells into the gel, and we put each DNA fragments into the different wells. And so what would this first well look like with this big piece of DNA? Chandler, are you playing a video game or you watching this?
I'm watching this.
What would this first piece look like if you put it into this well?
Just one big line.
It's one line close to the top because it's one big piece. What would this look like?
A line close and a line farther.
2 lines. Those are relatively equal in size. Be something like that.
Mr. Willis?
Yes.
She sent me your help.
Okay.
You want me to teach
Who's the best student? Right here. Josie, go draw what the other 2 would look like.
Oh,
Go, Josie.
Okay. So, 3 pieces. What do you think's gonna happen?
Just put the line.
Do some pointing.
So there's a bigger one,
Oh, nice.
and then a medium one,
The big one's gonna go with
and
the
then a
other
teeny
2.
tiny one.
Where's that?
So where do you think it'll be,
The
Chandler?
ones with the other 2.
Like here?
A little lower.
This one?
Like in between the big 2.
I just don't know if it's gonna work.
Like yeah.
big one?
I'd say
Right,
like,
right there. Oh, that's perfect.
and then,
I don't know.
What do you think?
yeah.
Yeah.
I don't know.
And then a
I
little-
disagree. I disagree.
And a little one close to the block? No, closer.
No.
You're
so
wrong.
You're wrong.
No,
And then these are about the same.
I disagree.
So
I don't agree.
I'm not giving you credit.
there's 4.
There's 4
These are the
segments.
same size.
They're 1 nucleotide difference.
Okay, there's a little difference.
There we go. Yeah, I'm happy.
Good job, Justin.
Okay, it was a group effort.
I love the 3 people clap.
Yeah.
I love them because one of them was me. So.
Thanks. That's really nice. I think your math drawing is going really well. My math is so good. Oh, I don't know how much does it even. If you get a thing. You're supposed to draw this map.
I forgot about that.
Really well. Here.
Oh yeah.
That's what
What's hard?
Yeah, because they can't handle Chrome at all, and my whole body hurts.
They can't handle Chrome?
Corona. They're like.
Did the other teams?
Like, not even people on my team tried to. Like, mine, I was one of the people and I was like,
The coaches did.
no, I didn't think so. And so I was like, you're stupid.
And they probably were around me.
Yeah, they were.
If she was at Thomas Club.
Oh, no, yeah. So, we're low to another competition.
Y'all guided me, so I don't want to hear it.
But then
I
also.
think it looks great.
I guess the third one.
Wow, great.
So she, we have to teach a whole new girl. And that one
If we get this
girl-
wrong, it's okay.
If you get this wrong, Mr. Wilson's never gonna trust you again.
I'm
You just call me a
really like-
Let's see if she got it right. Three pieces, 3 lines, 4 pieces, 4 lines. Pretty good. Give Josie a big hand for getting it. All right, y'all see how to do it? Y'all understand this? Why do the pieces go that way?
Negatively charged.
There's a positive charge on that and.
There's a positively charged electrode on this end. There's a negatively charged one over here. Yes.
Oh, sorry. I was very confused.
Oh wait, yeah, I got it wrong. There's a negatively charged electrode here. There's a positive here,
I was about to say,
and it goes toward the positive.
I was very
Good job.
confused on that.
Okay. Now there's a technique called Southern blotting. You might hear about or read about. What you do is you can cut up the DNA with restriction enzymes. And in this, sometimes they'll not even get just variable sequences. They'll cut the entire DNA from the whole cells up. And so when it runs down the page, it just looks like there's so many lines that they all run together. It just looks like one thick strand. Okay? And then, so how can you make any sense of that? Well, they take a sheet, this nylon membrane, and they lay it on top of the gel, and the DNA will like stick to this membrane. It's like a piece of paper, really. And you lay it on top of the gel, and the DNA will like stick to it. And then they put markers. They put DNA DNA probes are what they're called. A DNA probe is a short piece of DNA, and it's just half of it's just half of a DNA strand, and it will stick to the DNA that you're interested in. So if you put a probe onto this nylon membrane that is for a certain DNA sequence that you're interested in, they'll make the probe radioactive, or fluorescent, I should say, so that the probes glow. And the probes will be everywhere on this nylon membrane, but they'll only stick to the DNA that has the complementary sequence to what they're looking for. So they put these probes all over the place. If the complementary sequence is next to it, they'll stick to it and glow, and they'll light up wherever the correct sequence is. So you'll be able to see just the bands you're interested in. And so it's basically a way to find genes. If you're looking for a certain gene, and you have a probe that will stick to that gene, then you can take an organism's DNA, the entire DNA strand, you can cut it up with restriction enzymes, spread it out through a gel, and then release the probes onto it. It'll light up wherever the complementary sequence is. So, in other words, this one at the end has the genes there, this one at the beginning has the genes there, this one has the genes there, and it looks like this one doesn't, whatever's in that lane doesn't even have the genes. So this southern blotting technique is a way of finding genes within a sequence. There's another technique called northern blotting, which instead of using probes to find certain DNA sequences, you use the probes to find RNA sequences. You can run RNA through the gel and search for certain RNA sequences. And we call that northern blotting. We aren't going to be doing any southern blotting or northern blotting techniques in this class. But if you take a forensics class or DNA technology, you might do some of these things. Maybe you'll take something like that in college. Let me show you. I stumbled a little bit in trying to explain that. Let me show you a little video footage of it. This might help. Do you like video footage?
Yes, sir.
What? What is YouTube? I mean, now I can't even. Yeah.
Let's do another after
I can't even get to YouTube like in general. Okay, here we go. Southern blotting. Southern blotting.
Mr. Willis, do you still teach psychology?
Heck yeah, but not this year. We can't have it. We didn't have enough. I had to teach 3 biology, so.
The human genome contains about 30,000 genes. Therefore, in order to examine a single gene or a set of genes, we must first isolate a smaller DNA fragment. This is achieved by the use of restriction endonucleases. These enzymes cut DNA at sites with a strictly defined sequence of nitrogenous bases, forming fragments that contain different numbers of nucleotides. These DNA fragments are sorted according to length by means of electrophoresis. The location of a DNA strand after separation can be observed under ultraviolet or UV light. For further examination, the DNA is transferred from the electrophoretic gel onto a nitrocellulose membrane. This is referred to as the southern blot technique. In this process, a sponge is placed in an alkaline solution, and the electrophoretic gel, a nitrocellulose membrane, and layers of absorbent paper towels are placed on top of it. The sponge absorbs the alkaline solution, which is sucked up by the layers of paper. As the solution passes through the gel, the DNA is denatured, that is, its strands uncoil, and single-stranded fragments are washed out of the gel and fixed on the nitrocellulose membrane.
Well.
Morning, I am good.
You want to like program?
Yeah.
Would you rather do that instead of learning about cloning?
Only a little bit, just a tiny bit.
All right, let's look at cloning of a gene. How about that? Not quite programming, but. Let me remove these and remove this. here. This over here. And this will represent human DNA. And this will represent a bacterial plasmid. Do you know what a plasmid is?
The little loops.
A loop of DNA that you find in a bacterium. This is the plasmid. And they represent the plasmid with that blue circle on the right. So, what you can do, what we've learned through DNA technology is the ability to move genes from one place to another. Chaz, are you with me here? You seem like you're playing a game. Just make sure you're with me here, because this explains it really well if y'all would watch this. So, we have the ability to cut genes out of the human genome and put them in other organisms. They've done this for a lot of things. One of the first things that this was done for was to make insulin, have bacteria that can make insulin. A lot of people have diabetes, and they're allergic to insulin. If you have diabetes, you got to give yourself an insulin shot. Have you ever heard about that? We usually get the insulin from pigs and cattle. But they figured out a way to get the insulin from bacteria. And they can do it a lot cheaper. And it's human insulin, so no one would be allergic to it. A lot of people are allergic to pig and cattle insulin. That's normally where we get our insulin from before they invented this technique. But what you do is you take the human DNA and you use a restriction enzyme to cut the human DNA. Now, by the way, I don't know if I said this, restriction enzymes actually come from bacteria. Thomas, are you with me here? Restriction enzymes come from bacteria, and bacteria use them as a defense mechanism to cut viral DNA. If viruses infect a bacteria and inject DNA into the bacteria, the restriction enzymes in the bacteria will cut up the viral DNA, and it won't make the virus sick. This is how viruses protect themselves.
Because they don't have splices.
I mean, this is how bacteria protect themselves from viruses. They don't have spliceosomes?
Yeah, to cut up or take out. Never mind.
You're right, they don't. But that wouldn't protect us from viruses.
Oh,
But anyway,
that's true.
so we go through the DNA using a restriction enzyme that we stole from a bacterium. And we cut out a particular gene. Snip, snip. This comes out. Let's pretend this is the gene for insulin. This is the insulin gene. Now, I only have it as a few bases long here. In actuality, it's like 1,000 bases long. Yes?
I thought to cut it out, they had to match up with like the GAA TTC.
I'm going to show you that in a minute how it matches up. To cut it, you just cut at GAA TTC. So there's a GAA TTC sequence. Snip, snip. That opens up. Here's another GAA TTC sequence. Snip, snip. This opens up. So we've basically cut it out from the human genome. This is the human genome. You just take a cell from a person. You take a hair sample or something, and it'll cut out the insulin gene. And then you take the plasmid from the bacteria. You also cut the plasmid at GAA TTC. Snip, snip. The plasmid opens up. You then take these insulin genes that you've cut out, excuse me, you put them in a test tube with the cut plasmids. And you put in an enzyme called DNA ligase. You probably remember DNA ligase from way over here when we put together the Okazaki fragments. Remember that? Remember DNA ligase? DNA ligase will seal this, these are called sticky ends. This sticky end here, AATT, coincides with this sticky end, TTAA. And the DNA ligase will tie in this G with this A, and tie in this G with this A over here, and we say we've spliced the gene. Have you ever heard that word splice?
Mm-hmm.
There was a movie called Splice, where they made this alien person that had, like, lizard DNA. She went crazy and killed all the scientists. It was a ridiculous movie. But anyway, the DNA ligase here splices here and here, ties them together, and now we have a bacterial plasmid with human DNA. And this pink represents the insulin gene. You can then take the plasmid, put it back into a bacterium, and the bacterium can make insulin. And not only will the bacterium read this gene and make insulin, but the bacterium can copy itself and clone itself. And when the bacterium copies itself, it also copies the plasmid that's inside of it. So you go from one bacteria with this plasmid to two, and then 4, 8, 16, 32, 64. The bacteria just keep multiplying, and you can end up with millions of bacteria, and every one of them is reading this gene and making insulin. They grow them in these huge containers. It's industrial size. They have buildings bigger than our gym, filled with these containers, and inside of it are all these bacteria growing, and they're making insulin. And they got a little spigot on the bottom that has a filter, and the liquid from inside comes pouring out, and it's got a bunch of insulin in it. And they could separate that with big centrifuges and bottle it and sell it. And a little bottle of it sells for about $100. And they have millions of diabetics in the world that buy these things. And they're rich. And it's human insulin, because it's made from a human gene.
Why don't they just put the bacteria in
In the person? That's a good question. If you put bacteria inside people, that's an infection, and that can kill you, because the bacteria will eat your body while they're inside
What
you.
if it's a not harmful one?
Every bacteria has to eat, so they would all be harmful. Now, you do have bacteria in your gut
Yeah, that's what I'm
that aren't harmful to you, but if they were making insulin, that would probably have a hard time getting into your body, because they're in your intestine. You just poop out whatever they excrete. So I don't know. Maybe that would work. Maybe you'll be the one that does it. Probably wouldn't work, but that would be cool. If it did, you could get insulin that way. So this is called. We call this this little piece of DNA, this foreign piece of DNA. We say that it makes the organism a transgenic organism. Transgenic means it has its own DNA, and then it has DNA from another organism in there. They've done this for a lot of things. I was reading about one where they took a gene from a spider DNA, and it had instructions for making spider web. And they put it in a bacteria, and they're growing these bacteria that are making huge amounts of spider web material.
So
Spider web material is some of the strongest material on Earth. Did you know that? They say if you could weave enough of that material together and make a shirt, it would be this light and bulletproof. Imagine putting that on all your policemen and all your... heck, I would wear one around.
I would too.
Wouldn't you like to have a bulletproof shirt?
I would wear a mask.
What about lining cars and tanks with it and stuff? They'd be real light and real tough. I mean, this is the the possibilities are endless with genetic engineering. That's the thing nowadays. Learning how to do this, working at companies that do this, that's big money nowadays. If you're looking to get rich, this might be your thing. Learn how to do these techniques. Have you ever heard of Dolly the cloned sheep?
Yeah.
So how do you clone a sheep? This is called, whenever you're copying a gene, when we put this gene here and have the bacteria copy itself, we're cloning the gene. That's different than cloning an organism. When we're cloning a whole organism, we call it reproductive cloning. Reproductive cloning is cloning a whole organism. I kind of want to have myself cloned. Maybe an army of Mr. Willis's running around ruling the earth. That would be good for society, probably. How do we do that? We take an organism that donates a cell. I think they get them from the mammary glands of the sheep, and they remove the nucleus. So they pull the nucleus out. Now they have. Oh no, I'm sorry. This is not from the mammary gland. This is the egg itself. So they take the egg from a donor, and they remove the nucleus that was in the egg. So basically they have an egg cell that doesn't have a nucleus in it. Then they take the nucleus from another sheep, and they put it in place into the egg. They have little tiny syringes that can actually stick into the cell and squirt the new nucleus in there. Now the nucleus, of course, has all the DNA from this original nuclear donor. So this is the animal that we're going to be cloning. We take the cell, this is where it first from mammary glands. Take the cells from its mammary glands. You know, those are the glands that provide milk. Y'all know what a mammary gland is?
Mm-hmm.
Take a cell from a mammary gland, put it into this empty egg cell. Now it's got the nucleus from this animal. And then the scientists let that multiply inside a test tube. It will become a big ball of cells. And they will eventually implant that in a surrogate mother who will carry this growing sheep. This is what the ball of cells looks like, but it'll eventually grow, and this sheep will be pregnant and will give birth. And the sheep that it gave birth to was called Dolly. They named it Dolly. And that was an exact clone of this, because we took the nucleus from this. So that was the first successful vertebrate animal clone. And that since then they've done rats and dogs and cats and mice and all sorts of different animals. It's never done a human. You could theoretically do a human, but it took like 400 or 500 tries to get Dolly. Every one of them died before birth at some point. So you can imagine, since they're not that good at this, it wouldn't be worth to try it on humans if you kept getting these aborted fetuses and things. But I bet one day there'll be human cloning if they get good at it, where it works 95% of the time or something like that. They would probably do that, especially for couples who can't have kids. Maybe this is a new way to have kids. You could clone yourself. Have a kid that's just like you. You're like, I'm great. Clone me.
That's terrifying.
And you could raise yourself, in essence. Now, the last thing, really, in the book, again, this is a long chapter, so you've had 2 days to read it here. But it's gene therapy. So, in gene therapy, what they can do is they can take DNA that is good, a gene that they want to introduce into a person. So this has been done for some genetic diseases. So cystic fibrosis. Let's talk about cystic fibrosis. So in cystic fibrosis, you have two bad genes, right? Little c, two recessive genes that aren't making the correct protein that you need. The protein that you need is the big C, and that is a protein, a channel protein on your cell's membranes that transports salt back and forth across the membrane. If you have the non-working version, you don't transport salt correctly across your membrane, and it ends up forming thick mucus, right?
Mm-hmm.
That's thick and clogs up your passageways. What if you could inject the good gene into the body? They figured out a way to do that. They put the good gene into a virus. They removed the viral DNA first. I mean, you don't want to infect somebody with viruses. But they removed the viral DNA, and then they put the good gene in that you need to make the correct protein. They put it in the virus, and then they inject you with the virus. And the virus goes into your cells. This particular virus goes into your cell and hooks up with the nucleus of your cell and injects the good gene into the nucleus. And now the correct gene is in the nucleus. This cell will read that gene, will make the correct protein that will go on the surface of the cell, and hopefully correctly transport salt through the cell, and you won't make the thick mucus. Now, they're running into problems with this. For one thing, you have an immune system that attacks viruses, right? And so what ends up usually happening with this is it works for a while, and then your immune system starts fighting that virus, and the virus won't be able to get into the cells anymore. Do you know anything about this type? Have you ever run into this type of therapy?
The current medicine I'm on, Trikafta, is something like this.
Oh, is it really?
Mm-hmm.
Okay. Does it help?
For most people it does.
Yeah.
It just kind of keeps me at a constant instead of a fluctuating.
Oh, okay. Well, again, this is kind of the future of medicine. This genetic engineering, and there's all kinds of genetic diseases out there, genetic disorders, and if we can figure out how to get good genes into your cells, we can get rid of a lot of those. So make sure you read this chapter, all of 17.1. There's some stuff about transgenic organisms. They've got transgenic plants, animals, bacteria. Read all about those. Probably be a quiz tomorrow.